So today's, it's not really a lecture, it's much more of a discussion of various things that we have done in the lab or I have been part of. And to say that it's really important to test assumptions that we are making a lot. Anytime we write any model, anytime we're doing any experiment, we are making a lot of assumptions about what the data are, what kind of data are we getting, how do we interpret it, under what conditions can it be generalized to something else beyond the work that we're doing, right? So can somebody tell me some assumptions that you can already see are being made in what you've learned so far in the course? Constant population size, all right. Well-mixed populations, right? There's also, although Mike is talking about where there is spatial structure and what happens, right, outside of that. But okay, so some assumptions are being tested and being pushed the boundaries for. Any other assumptions that you've encountered so far? Sorry, what? Constant mutation rates, you mean not changing over time? Okay. Migration is an assumption, like migration is happening, or no migration, or constant migration? Okay, all right. Yeah. Time scale is maybe the same, but maybe not the same in reality. Sorry, the other way around. Yeah. Okay. Yeah. Assuming random mating. Yeah. Assuming loci are independent of each other. To some extent, you've already broken, right, these assumptions or tested, pushing them a bit. Right. So we make a lot of assumptions. Anybody want to say some experimental assumptions? What are we when we do it, or when you're collecting empirical data in the field, perhaps? Some examples of assumptions you might be making. That there's no bias in your sampling itself, but it's not truly an assumption. Ideally, you should be thinking about it and trying to make sure it is true. But yeah, okay. Any other assumptions that you've encountered? Sorry? The samples you're collecting, for example, for your field data collection, may be biased and not representative of the population. So that's an assumption. Even if it's a larger size, if you're still collecting from one corner of the, I mean, yeah, ideally you should be doing that, but still. Okay, so the kinds of assumptions I'm going to talk about today, I'll give 3 examples. One is from an experimental assumption from a protocol that is used a lot in experimental evolution. Second one is about whether synonymous mutations, what is their impact on fitness? And the third one is about what is the DFE looking like? How many beneficial mutations, how many deleterious mutations are there? And so some of them are more specifically related to parameters which you'll be encountering in some of the models that people are talking about. And some of them are much more, if you are doing work in the lab, something to be aware of how something that's taken for granted may have nuances that are important to consider. So as always, ask questions if something is not clear. So I'll start with this one, synonymous mutations are neutral. You've heard this sort of implicitly in several areas of work that are being done in population genetics, that you are, or in evolution more broadly, that synonymous mutations are a good baseline for us to ask about what is the rate at which mutations are accumulating, at which substitutions are fixing in populations. Then we can compare that with genes or regions of our interest to say, okay, is that region evolving under selection likely or not, correct? So the neutral mutations are thought to be represented well by synonymous mutations. This has always been the case for a long time. Other kinds of regions in the genome are also considered to be good representative, or as good as we can get, of neutrality, right? But I'll focus today on synonymous mutations. So going back to this kind of plot that I showed you earlier in my previous lecture, here is the type of selection, evidence for selection that people have thought about, right? So we went over this for mutations or genes or genic regions in general. And now I want you today to focus on the pink stuff, right? This is where some of the data really support the idea that it's not always neutral. Synonymous mutations are not always neutral. So for example, in genome-wide association studies, particularly for human diseases of various sorts, people have found reasonably strong indications that allelic variation, which is synonymous, actually is associated fairly strongly with the phenotype of interest. And that effect size of that association is sometimes not very different from what you see for non-synonymous alleles, right? Similar things are found in experiments in vitro as well. So you take proteins where there's a hint that a synonymous variant in the population that's segregating may have a role to play. And then you make that synonymous change in a cell line in a petri dish. And you're able to see that something changes about the way the protein folds, the way the protein is moving to different parts of the cell, the way it acts if it's an enzyme, and so on. And that those phenotypic effects are sometimes not very different from what they observe for non-synonymous changes, right? So it's easier for us to believe or understand that non-synonymous changes may have phenotypic impacts, but there are several instances where we know that that's the case for synonymous changes as well. Similarly, in evolutionary experiments, synonymous changes are sometimes found to increase in frequency, like what you'd expect for something that's under selection, that's driving adaptation potentially. I'll show you an example of this from work that I had done in my postdoc and then continued here. If you look at a set of mutations that you make in a cell, in a lineage, in a protein, and you then ask what is the impact of those mutations on fitness or phenotype, you again sometimes find that synonymous changes do have not terribly different impacts compared to non-synonymous changes. Looking at this a slightly different way, looking at the distribution of fitness effects, again, I'll show you some, did I include that? Maybe I didn't include it. I'll send you a paper that has looked at these kind of DFEs for synonymous and non-synonymous substitutions and asked what happens to the DFE. And finally, when there are cases of evolved adaptive mutations where you can show that that one mutation that's synonymous has the ability to increase fitness in that environment, then that's sort of really, really clear that that is what's going on. And I'll show you an example of this again. Yes. Can you speak up a bit?
Was that the data?
No, that was just an illustration. Yeah, that's a cartoon to get the point across, and I'll show you some real data. Okay. All right. So the first story I'm going to tell you today is about testing fitness effects of codon usage. So this is the only situation where, in the examples I'll give you this time, where I wanted to test this assumption, which I encountered again and again in literature, right? So towards the end of my PhD, where I had no idea about molecular evolution much at all, I was seeing all these papers that talk about how synonymous changes are neutral, and that's a nice background to compare for cases where it may not be neutral. And on the other hand, I also came across a bunch of papers that showed that if you look at which codons are being used to encode specific amino acids across the tree of life, there are very big differences in that, and there are very strong biases, right? So for a particular amino acid like leucine, you can encode it in 4 different ways using 4 different codons. And each of those is used to different degrees in different organisms, in different genes, and so on and so forth. So why is there this bias and what's going on, right? So this is what drove me to ask, okay, can we experimentally test this? Because I couldn't find many experimental tests of, okay, so if I make a bunch of codon changes, does it matter at all, right? Let's measure it. So I chose to work with this bacterium called methylobacterium, which is a facultative methylotroph. It's not important what that means. Point is that it has this one enzyme called formaldehyde activating enzyme, which is very important for its ability to use methanol as a carbon source. So many of you who do experiments would know that ethanol, methanol are rather toxic for cells, right? Why are they toxic? Because they get converted into formaldehyde, and that is what is actually toxic to the cell. So FAE has the ability to detoxify formaldehyde. It takes formaldehyde and converts it into other compounds, which can then be used to produce biomass. So not only can it detoxify methanol, we can actually use it to grow, right, as a carbon source. So FAE is an enzyme that is fairly conserved amongst closely related taxa in this group. And if you delete it, these cells are not able to grow on methanol. Okay, so that's a nice, simple phenotypic fitness assay which we can have. Are you able to grow on methanol or not? And how well are you able to grow is a quantitative measure of how well is FAE activity represented in the cell. So created different versions of this FAE, and then I'll show you the data in a minute, but basically found that different versions of this FAE gene with different synonymous codons, only synonymous changes, had different fitness, okay? So then these different strains which had different fitness, I evolved in the laboratory to ask how would they fix this problem of having poor codons, okay? All right, so here are data that show you the evolution experiment, but first here is what they started off with, okay? So here are all my different variants. Wild type is in gray, which is shown here by the dotted line. And I had versions where only frequent codons were used, right? So every codon in this 200 odd amino acid protein sequence was a frequently used codon, which was most highly used in the genome of Methylobacterium, all right? So the idea is that this should be the best set of codons to use, right, for each amino acid. And as you can see, that was really, really not the case at all. So I was like, okay, I'm sure I've done something wrong with this cloning, right? This is the first time in my life I'm ever doing PCRs and cloning. I must have done something wrong. So I made this again and again, find the same thing, right? So whenever you have a version of this gene, which is only composed of frequent codons, it really terribly does terribly. It's almost dead. It's as good as the deletion mutant. On the other hand, if you have this orange one, which is where all rare codons are used, so 99% of the codons in the cell are rare, which are not otherwise highly abundant in the genome, you see that the growth rate is lower than wild type, but still not as bad as this one, right? So the rare codons, all rare codons are actually a little bit better. And then there are all these three versions where half the codons are rare and half are frequently used codons. But the position in the gene is different, right? So in some cases, I've used rare codons that conserve residues. Conserve meaning across different Methylobacteria, which residues of FAE are conserved versus not. And one strain where the variable residues are rare, and this one where randomly half the residues are rare and half are not rare, okay? And then this one is an interesting one where only the active site residues are rare. All the other residues are frequently used codons. And you see it behaves very similarly to the one where everything is frequently used, correct? So this is the starting point. And then I did serial dilutions on a daily transfer cycle, allowing them to slowly develop the ability to grow on methanol. For these two strains, because they were dead in methanol, I added a little bit of another carbon substrate, which does not require FAE to grow, right? So this allows for a more gentle introduction into methanol, and over time I reduce the methanol, increase the methanol concentration and reduce the succinate concentration. That's the other resource that I used. So then I could have some evolution happen, and you can see in every case there's a large increase in fitness fairly quickly. The different lines are showing independent lineages that evolved on their own. And you can see that the increase in fitness is very rapid for these two populations. Notice within 70 generations, they're already sort of plateauing. They're not increasing anymore. Whereas these other lineages, which had fewer rare codons and fewer frequent codons, they took a little bit longer to get to whatever is their plateau point, right? And then the question, of course, is, okay, how are they fixing this problem that they had originally started off with? So just to summarize the data I've shown you before, here's the initial fitness. So here I've averaged across all the different lineages for each strain. And here is where they got to, so you can compare, right? So the red and the pink, the pink and the black are very interesting because they get almost as good as everybody else, starting from a very low point and starting from really kind of being dead. Okay, so how did this happen? In triangles, you see cases where there were single synonymous or non-synonymous substitutions that I found when I sequenced these genomes, right, in the FAE gene. And I put those single nucleotide changes back into the ancestral genome. So that is what the triangle shows. The ancestors are shown in circles here. And we also measured how much protein is being produced in the cells, the FAE protein, which there was a marker, a tag on it, a his tag. And we also measured how much enzyme activity there was, okay? By extracting the protein, this is work done by a collaborator, I don't know how to do this. Extracting the protein, putting the substrate in, asking how much of the final product is being made per unit time, et cetera, right? That's what this axis is. And I measured the growth rates of these, and you can see that here's where the strain started off. Here is the effect of the single point mutations that I observed in these strains in different lineages, right? So the pink lineage got 3 different mutations. One of these was synonymous, 2 were non-synonymous, I think. The black one got only 1 synonymous mutation, again and again and again in all lineages, right? That was the one amazing synonymous change that fixed things, increased growth rate, increased protein production to some extent compared to the ancestors, but not by much. And here are all the effects of other mutations that were found in the other strains. Most of these were non-synonymous. I think one of the blues is a synonymous mutation as well, right? And then as growth rate increased, typically enzyme activity also tended to increase. Here's where the wild type sits for comparison. So what's also interesting is the growth rate increase is much higher compared to what you would expect or predict based on the enzyme activity, right? So the wild type enzyme activity, we think, is much higher than what is actually necessary for growth in methadol at that concentration that we typically grow in. So what does all of this show you? All of this shows you an instance where there were large deleterious effects of synonymous changes that I made earlier, right, initially. It also shows you that all of these changes that were deleterious could be compensated by single substitutions which were either synonymous or non-synonymous. Shows you that this was very repeatable across different lineages. And these data show you that these synonymous changes as well as non-synonymous single substitutions can have enormous impacts on not only growth rate, which is our measure of fitness here, but also the actual enzyme production or the enzyme activity. Now, if you ask me why this is happening, I have no idea. Yeah. The one synonymous change that you mentioned, no, they're different for each strain. Thank you. That's an important point to bring up, that the mutations that are compensatory for each of the strains are all different. There is no sharing of substitutions across strains. So clearly the strain background also determines what kind of mutation is going to be beneficial or not. Right? Any other questions? Yeah, the changes that we made in that strain, in the FAE strain. Everything else in the strain is the same. So the other genomic background is identical. Okay, all right. Okay, so then here is some work that had been done before, comparing sequence data in Drosophila. This is by Hiroshi. And Hiroshi found that you can actually infer weak selection at synonymous sites in the Drosophila genome if you start comparing rates of substitution, rates of synonymous and non-synonymous substitutions at these sites. And so he had this paper earlier, which I was very happy to see because we based some of our work on what he was finding. But he was suggesting that there's very weak selection, right? If there is selection at synonymous sites, it's quite weak. So in 2014, which was the first PopGen school, Hiroshi was here. And so we had a mock fight because I was saying, no, selection can be very strong. And he's finding very weak selection throughout. This is just for fun. Anyway, so since then, I want to show you a few examples of other studies that have also shown results for Drosophila. I really like this particular paper. So here they looked at different species of Drosophila, different populations of Drosophila. This is, for example, populations from Zambia, and this is the DGRP. This is a lineage or a collection of lines people have created as a sort of reference material for Drosophila, right? Where they have sequenced everything, the lines are inbred, and there are many, many lineages available for people to then use for all kinds of things. So what they see here, what they're plotting here is here's a full data set for how much polymorphism you find at 4-fold degenerate synonymous codons. What this means is you're looking at amino acids where all 4 possible substitutions in the last base is going to lead to the same amino acid, right? So there are a few such amino acids in the genetic code. You look at only those where there's 4-fold degenerate. Each of the 4 substitutions gives you the same outcome in terms of amino acid. And you look at short introns as another region in the genome, which may be evolving under weak selection, right? Which may be an indicator of neutrality. So comparing these, you can find that in both of these populations, the 4-fold degenerate codons have slightly less polymorphism compared to the intron sites, right? Similarly, if you look only at preferred codons, look at non-preferred codons. In preferred codons, the signal is a bit stronger, and unpreferred codons, that signal disappears, right? Which suggests further that this is something to do with which codons are being preferred in particular locations. You also find that the polymorphism ratios are nicely correlated. with the relative synonymous codon usage, right, in the Drosophila genomes. So as codon bias increases for a particular preferred codon, the polymorphism at that site between, for those codons between introns and fourfold degenerate sites is also increasing, right? So again, this suggests, these data sets suggest that there is something specific about which codons are preferred and which ones are not preferred. There's another study which I forgot to put in here, which is also one of my favorites. This was a much older study with Drosophila, where David Carlini had taken ADH gene of Drosophila, which is the alcohol dehydrogenase, very important for larvae to be able to survive because it allows them to metabolize ethanol, which is produced in the media, right, as they're eating. So ADH gene, he changed a few synonymous, made a few synonymous changes, and he found that the larvae were now producing less ADH and were not as likely to survive, right? So that was the only other experimental study that I could find which clearly showed that there may be really big impacts of synonymous changes, but there was not much follow-up on that since then. Okay, good. So here's another case with viruses. This is also, again, something I really like. So this idea has been around for a while now, that you can take, if you want to basically make viruses which are not so good at infecting their hosts, right? Maybe one way to do it is to put in so many synonymous changes that they cannot revert back. But the large number of synonymous changes are a way to effectively reduce virus fitness, okay? So people have been trying this strategy in different viruses and testing it with animal models and so on. There's one paper which was very famous a few years ago, which showed this with polio viruses, where they made strains of polio virus, attenuated ones, but and infected mouse models with it, and showed that if you made a whole bunch of synonymous changes, the strain was not able to infect as well. And that's like serious drop in infectivity was useful because it could now be used as a strain to vaccinate people, right? Because it's attenuated virus. Okay, so here is one other study. This is by JT. Oh, my citation has disappeared. Okay, this is by Van Leeuwen et al. From Holly Wickman's lab. I can send you the citation of this paper. So here what they tried to do is ask, okay, which parts of a virus may be more susceptible to synonymous changes and the problems that arise because of synonymous changes. So they took, this is the phage X174, which infects bacteria. And they changed took different regions of the genome in which they introduced more or less synonymous changes, right? So that's what this plot shows here. Here is what they're showing where they're changing. And so this is a host that, the host for this phage is E. coli. And of course, the idea is that the phage is using the E. coli machinery to express its proteins and express its genes, right? So it has to match reasonably well with the E. coli codon usage if it has to effectively do this. So they took these data for which codons are used more frequently in the E. coli genome, looking at either all E. coli genes or the highly expressed genes alone. And you can see here, by the way, that the highly expressed genes are much more skewed towards using certain codons compared to others, compared to all other genes. And then they changed the phi X174 wild type, de-optimizing it deliberately to match different areas in this gene based on these data. And then what they looked at is what happens to the fitness of the phage in terms of infecting the E. coli host. Here's all the different genes that they manipulated. And here again, you can see that here's the wild type fitness. That's the baseline that they're using. So about 20 plus phage particles are produced when it infects E. coli. All of these guys have low fitness, right? So here in this case, you see that de-optimizing the codons reduces fitness systematically. But here as well, there isn't a very strong correlation with how much you de-optimized, right? Or are there specific codons which are much worse to de-optimize, much better to de-optimize? So in general, this is something that is very useful also potentially and has applications, the fact of using synonymous changes. I want to end this part by giving you a sort of overview of the history of thinking about synonymous changes in an evolutionary perspective. So this is part of this. I feel like my bottom slide of the slide is gone. Okay, anyway, this is part of a book chapter where I tried to trace this history. And so initially, the prevalent idea was that synonymous changes are neutral because it was observed that synonymous changes, there are many more of these, right, in populations when you look at what kind of substitutions you find. you find many more substitutions at synonymous sites compared to non-synonymous sites. A large fraction of mutations evolve effectively neutrally. This is part of what Kimura proposed with his neutral theory. You've heard this before. People then started finding some indirect bits of evidence to suggest that codon bias may not be indicative of neutrality. Codon bias may be evolving under selection. So there's some codon usage differences across genes that were found. People also found that codon usage differs across genomes. I already told you before, codon usage and tRNA abundance seem to be correlated with proposed a mechanism for what might be going on, right? That if you use some codon more, then the tRNA that recognize that codon should also be used more in the cell, right? Or has higher copy numbers. That was observed. People found that functional genes have lower synonymous substitution rates compared to pseudogenes. Pseudogenes are genes copies which are no longer functional, and they can accumulate and evolve more neutrally. They can accumulate mutations. People also found that if you introduce rare codons in yeast, this study is with yeast, reduced mRNA and protein levels, and so on and so forth, right? So, this study is important or interesting that synonymous mutations were fixed repeatedly during experimental evolution with phage. This is work from Jim Bull. And so this suggested that something is going on, but then it was kind of ignored, right, for a while. So fine, so then later on, more recently, we've had much more direct evidence that synonymous changes can have long-term evolutionary effects. So some studies here in the 2000s starting to show that deleterious codon changes can be rescued by regulatory mutations. This is work from a group in Spain, showing that antibiotic resistance can be modulated by synonymous changes in the resistance gene. There was a paper from Susan Bailey showing that synonymous mutations show up during adaptation in Pseudomonas in the laboratory. This is my work that I just told you about. And then there's a very nice paper showing that globally rare codons are deleterious. This is where they did a genome-wide sort of mutagenesis study and showed that these can be rescued by tRNA overexpression. This is in E. coli, right? So there's again a hint back of synonymous changes coinciding with codon shifts, which are important. So we have reached this idea where it seems like, and what I'm showing you here is just the studies that talk about evolution in some shape or form, right? There's a whole other literature, a sort of medical work showing for human diseases, for animal diseases, there's a large number of, there's a collection of studies showing that there's associations between synonymous changes and phenotype or disease. And there's a even bigger literature in molecular biology showing that synonymous changes can have impacts on protein structure, protein folding, mRNA degradation, transport of mRNAs and proteins in different parts of the cell, and so on and so forth. Right? So it really is all over the place, yes. In some cases, yes. So there's good evidence showing that if you introduce rare codons in sort of random places in your mRNA, then the ribosome machinery sort of stalls there, right? And that explains the deleterious nature of rare codons in some cases. But in other cases, like in my study that I showed you, the rare codons are actually better than only frequent codons everywhere, right? So there's another hypothesis and some support for this suggesting that you actually have rare codons that have evolved at specific positions in a genome where a pause is actually beneficial for correct folding and movement of things. So the point I want to make actually is that the mechanisms are extremely diverse, right? And they are very, very different across different species. They are different across different proteins, different across different contexts in which you're measuring fitness of phenotype. So there isn't a single solution to this problem, which is partly why I have stepped away from it a little bit, because it's just a bit maddening to say, okay, why is this happening exactly, right? But it's fascinating that now as people study more and more in different organisms, that they keep finding instances where synonymous changes or codon changes are important for fitness. Usually deleterious, but sometimes beneficial as well. Yeah. And so I want to I think, end this part saying, okay, synonymous mutations are not always neutral, but many big questions remain. For example, we don't understand, why or how are synonymous mutations not neutral when we do experiments like this. And yet, in natural populations, there are many more synonymous substitutions than non-synonymous substitutions, right? So what is going on? And so one suggestion from a review done, a meta-analysis done by Susan again, which is what I forgot to put in here today, is she looked at the distributions of beneficial fitness effects for different mutations. So people have looked at synonymous, non-synonymous changes and so on, and asked, what is the magnitude of the beneficial effects that are found, right? And what she finds in the meta-analysis is that many times the magnitude of the beneficial effects is a bit lower than non-synonymous mutations. And so the suggestion is that, therefore, synonymous changes are less likely to fix in populations, to be promoted by positive selection in populations. And the deleterious effects are also weaker, which means they're able to hang out for longer in the populations. And maybe that explains this weird dichotomy that there is a lot of synonymous substitution compared to non-synonymous at long time scales in populations. And yet in short time scales and small experiments that you do in the laboratory, you will find big effects of synonymous changes. Right? A lot of work can be done to address these big questions, and I hope that one of you will take this up. All right, good. Second part that I want to move to is thinking about mutation accumulation experiments. Yes? No, not necessarily. Yeah, there are certainly fewer studies of experimental evolution with eukaryotes compared to prokaryotes that address this problem, but nothing pops out as being very obvious. Anything else about? All right, good. So the second assumption I want to address today is mutation accumulation experiments and the idea that they remove selection, right? Or they suppress the action of selection as much as possible. That's still true, and that still happens to be the best way that we can do this, but I'll show you that it's not entirely the case. So I think in some lectures before, you must have heard this, that if you want to introduce or if you want to reduce the activity of selection in populations, you need to evolve them under very small population sizes. The easiest way to do this is have them go through bottlenecks, right? So this is a protocol that is followed, especially in microbes very often, where you take agar plates where you can spread out your bacterial cells, and you mark a colony before anything grows, and that is your random choice of which colony you're going to use to propagate next day, okay? So basically what you're doing is, because each colony is coming up or developing from a single cell originally, the single E. coli that happened to land there in that spot, you're allowing for single cell bottlenecks to appear again and again and again in the population. And this should effectively reduce the effect of selection, because it's random which colony is used to propagate the next day. And there is relatively little time in which there's any growth happening in this colony. So competition and all of its effects that could act to give impacts of selection are low. Okay, so an example of this is what is observed in Mueller's ratchet. So these are data from viruses infecting a plant host. And here, yeah, I'm sorry, all of my citation bottom part is gone. So I'll send you the papers later. So here what they did is they did serial passages similarly with bottlenecks where viruses were allowed to infect the plant host. And here is the Malthusian parameter, which is the growth rate of these virus populations over time. And as the bottlenecking proceeds, you see that the fitness is reducing, right? And so this suggests that, as you would expect under repeated imposition of genetic drift over time, where deleterious mutations can keep accumulating over time, you will lose fitness as these deleterious mutations accumulate. Here's another interesting study where they took C. elegans, these are worms, and allowed them to undergo bottleneck serially, but they maintain different bottleneck sizes. So here is a bottleneck size of 1. So single worm is allowed to reproduce every generation, every transfer, whereas here there's 10 worms and 100 worms. Right? And the idea is that as you increase the size of the transfer bottleneck, you are allowing more and more selection to act and more drift to be dominant, right? And what you see in terms of mean productivity, which is how many offspring these worms are producing per capita, you find that across generations, the bottlenecking of the very severe bottlenecking leads to a very sharp drop in fitness, whereas the more relaxed bottlenecking doesn't, in this time frame, have a big impact on reduction in fitness, right? So suggesting that mutation accumulation is a effective and good way to impose evolution by genetic drift and reduce selection. So we were doing some work with Lindy, who is a population geneticist and who's been in the school before teaching, to understand what's happening with mutation accumulation lineages in the lab. And here's a cartoon showing what might be going on to start pushing the assumption that, well, is it that there is no selection or very weak selection? How can we measure how much selection there is during this phase of colony growth? So imagine there's a green mutation that occurs early on during the growth. We're assuming the colony is growing from the center outwards, right? And that green mutation, because it's slightly beneficial, okay, allows slightly faster replication of the cells which have it. And that leads to what this pattern, this kind of pattern in the growing colony where that sort of spreads locally, right? It's of course not going to spread everywhere else in the colony, but it's just because it's producing slightly fewer offspring, slightly more offspring, there's a fanning out of this lineage. Whereas in a deleterious mutation case, with this purple mutation that may occur here, it will be lost from the population because others around it will overtake it, right? The wild type in this case. So what happens as a result of this is possibly that the green mutations are overrepresented when we take a piece of that colony and spread the next day, right? So that's what's shown here. Whereas the purple mutations are more likely to be not represented, correct? So when we look at the DFE, which we're measuring as a result of these single mutations that we collect at the end of that bottlenecking transfer period, the DFE may be skewed towards more beneficial mutations being represented in our final set of single mutants that we collect, right? So if we do the MA lines for a long time, we collect all these lineages, measure their fitness, we might see a DFE like this, whereas the actual DFE should be corrected towards the left because we're missing a bunch of deleterious mutations that actually occurred, and we're overrepresenting the beneficial mutations just because they're more likely to be there in the population in the end. So Lindy wrote equations to try and understand and model how this is happening. So here's exponential population growth. You've seen this already. The rate at which new mutations occur, the mutation rate is mu. N0 is the initial time point, initial population size in the colony, which is 1 because it's single cell bottleneck. And that's just the exponential growth. The growth rate of the mutant lineage then is given by R times 1 plus S, where S is the selection coefficient. And the size of the mutant lineage then in time tau, which is after number of generations of growth, is given by this, where you have tau minus t, which is the time elapsed for the colony to grow, right? And so then you can calculate what is the relative bias imposed by selection. How many, that's the rate of occurrence at time t of the beneficial mutations. Lineage size at tau for a given selection coefficient. Remember, the selection coefficient is going to determine how much bigger that fanning out of the green mutation is going to be. And the rate of occurrence similarly for something which is neutral, where the selection coefficient is 0. And this is what's assumed for everything else that's there in the colony. So you simplify that and simplify it further. If mutation rate is very low compared to growth rate, which is going to be the case for these E. coli populations, growth rate is very, very high. Mutation rate is still on the order of 10 to the minus 8, minus 10, and so on. So this is a simplified version that you get. This is the relative bias that's due to selection in the colony. And you can use this to then correct for your DFE, right? So you have your bins of mutational effects that you get in your N, in the DFE that you get in the end. And you can bin those mutational effects to say, okay, for this bin, I actually have an overrepresentation, like I showed you in the previous cartoon, of everything with that selection coefficient. In reality, it is not that high proportionally compared to the rest of the DFE. So you correct for that bias. And the raw versus the corrected DFE would look something like this. You weight the fitness bin in that histogram given the selection bias, and this is the correction that we do. And then you get the corrected frequency of each bin in the DFE histogram by summing it over all of the bins that you have in the histogram. So here's what you actually see if you do simulations accounting for this selection bias. So here's the relative bias, here's the selective effect S. You can see that as the number of generations increase, the amount of bias due to selection increases. That makes sense, right? If you allow a colony to grow for 3 days versus 1 day, you're going to have a bigger impact of that selection coefficient, the beneficial mutation in the 3-day time period compared to the 1-day time period. And that's what this is showing. The relative bias and selective effect interact in this way. The observations are, if you have selective effects, this is what the DFE looks like. So the true DFE is this black DFE, which as you see is left shifted compared to this no fitness effect line, which is in black. And as you have longer and longer periods of colony growth before you do your transfers, you get a more and more right shifted DFE, right? So you're more likely to see beneficial mutations in your DFE, and this is what the inferred DFE then would look like. So this suggests that then that anytime we're doing mutation accumulation experiments where there is a period of growth that's allowed between bottlenecks, you must correct for this effect. Otherwise, there could be a substantial bias in the DFE that you get. So we tested this with data which we had in the lab for E. coli again. So with 5 generations, 15 generations, 27 generations. These are all simulations, sorry. But these are matching observed DFEs that we actually see in the lab. So the purple is the observed DFE, the blue is the corrected DFE, and the black is the true DFE that's there in the simulation. And if you look at the fraction of the DFE, which is beneficial mutations, which is in blue, the gray is the neutral, and red are deleterious mutations, you find a very big impact of the correction on what these fractions will look like as you have increasing amount of time that's allowed for growth between transfers. And so just to summarize this part, if you have the fraction of DFE that is beneficial reduces when you correct the DFEs for this bias, and the mean selective effect of the mutations that you've encountered also reduces when you do the correction, right? Okay, and what was nice is actually people who were in the PubGen school some years ago, Anjali Mahilkar and Supreet, along with others, they found very parallel result with a different model that they set up for this kind of colony growth. And they also tested it in the laboratory with yeast, I think, looking at the fraction of beneficial versus deleterious mutations. Without selection, it is higher. With selection, it drops a little bit, right? And so it was nice that parallelly somebody else also had very nice similar results. All right, so mutation accumulation experiments are used very frequently to remove selection. In the case of bigger organisms where you don't have to wait for it to grow big enough to transfer, this is not such a big problem because they don't have to grow for a while before you do the transfer. But for microbes, bacteria, yeast, anything else that you can't see easily when they're in small numbers, this is going to be a problem and we have to correct for it. All right, the last thing I want to discuss is the idea that most mutations are, sorry, any questions about this previous part? Okay, last part is the idea that most mutations are deleterious. This is something that one sort of thinks intuitively makes sense, given that organisms have been evolving some environment or the other for a while. Surely they've gotten good at it reasonably, and there are therefore fewer deleterious mutations, more deleterious mutations that are possible, fewer beneficial mutations possible. But in fact, whenever people have tried to start measuring what is the fraction of beneficial mutations, even with cases where selection bias should be low. So this is with Arabidopsis or yeast populations. People have found these are a small sample of other studies that exist showing these data. You see that there are many more mutations that seem to be beneficial in all of these cases, right? And this has sort of been puzzling for a while. People don't necessarily have very good answers for why we see this, because the intuition is that this distribution should be far more skewed towards the left side, right? There are many more deleterious mutations that we should find. This is also the case in data that we had. So these are data from E. coli. We've done the correction using the bias correction that I just showed you in the previous set. So the blue ones are corrected and the raw DFEs are in gray. You find that in many different carbon media where we tested the effect of these about 80 to 100 mutants, single mutants accumulated through MA lines, we find that there are a substantial fraction of beneficial mutations that we see in every case, right? And this is not correlated with the ancestral fitness in those environments. It's not correlated with whether E. coli has evolved to use any of these resources in any obvious way. And so we don't really know what's going on. All right, so here as well, many questions remain. I don't have answers for this at all, why we see this exactly. I can suggest a few hypotheses that we have thought of and reviewers have suggested as we send this work out for publication. So one suggestion was that maybe we're measuring fitness here using growth rates, in our case at least, and growth rates may sort of have much higher fitness consequences, whereas if we measure fitness using other metrics such as comparative fitness, where our strain has to compete with a reference strain, and we measure fitness that way, where growth rate contributes a lot to that measure of fitness, but it's not the only contributor. Maybe in that case, we'll find a more compact scale of what the DFE looks like. Yeah. No, yeah, so they were all over the place. They occurred randomly in the genome. And when we look for our coding mutations more likely to be beneficial or deleterious, we don't see very big patterns like that. Yeah, are they coding or non-coding, synonymous, non-synonymous? Yeah, these data also support the idea that synonymous changes are not any different, significantly different from non-synonymous changes in what we see in our data set. Yeah, any other questions? Right, so one hypothesis is that the fitness metric that we're using makes a difference. Maybe the fitness metric we have, which is growth rate, is more likely to give us larger benefit mutations. Yeah. How many generations for what? Generations, I'll have to think back, but about 100 days of evolution to get one mutation. Yeah. So it differed for different lineages because we picked up lineages at whatever point they happen to have one mutation. So the answer is different. It's not the time span, no. We haven't explicitly tested for that, but I don't think so because we had sampling of mutations from all over. And yeah, but we can go back and check. Yeah. So protein structure will not change, right? By definition, synonymous changes cannot change protein structure because amino acid doesn't change. And that's the only thing that goes into your protein structure models. No, no. Folding can change if there's co-transcriptional folding, and that is known. But the folding doesn't change because of mutations in the protein itself. It changes more probably because of the time that the protein has to fold at a certain position in like a particular domain or something. So, but that effect still comes from the mRNA, not the protein. Once it's amino acids, then yeah, you can't change it. Okay, good. And so we don't know why we see very different outcomes of what's the fraction of beneficial mutations in different studies. Most studies find there are more deleterious mutations. Some studies find there are more beneficial mutations than expected, and we don't fully understand why. So this is also a mystery that hopefully some of you will try and solve as well, right? I think I finished way early than I expected, and you guys didn't have questions, so I'm done. But maybe I'll talk about just a couple other things in terms of this course and how you guys are doing and thinking about it. I hope that you're a little bit lost somewhere for some of the material that you're encountering. And that's fine, and that's good, I would say, right? Do what you can in terms of getting stuff out of this course, but push yourself a little bit at least, right? Because it's when you're pushing in your outside of your comfort zone that you'll learn something. The second point I'd like to make is, I think many of you are people who do experiments or collect empirical data or are not as theoretically oriented, right? But don't be afraid. I mean, clearly you're here, so you're not already afraid. But I would strongly suggest trying to understand as much of the theory as you can. Some of the really interesting stuff that me and my lab have been able to do is in conjunction with a theoretician, with Lindy. And that is really enriching when insights from both theory and data can be combined and brought together. So if you end up doing science in future in your lives, and you are one side of this sort of spectrum, try and find a great person to work with who does the other thing if you're not the person who can do both. So I find myself incapable of doing theory myself, but I want to understand what insights a theoretician can have, right? And that's really important and really helpful for me to do better experiments, for me to think about what gaps exist, for me to have explanations that I may not think of, but somebody who has that other perspective will think of and suggest, right? And so that back and forth is really nice and really useful. And hopefully you at least get a flavor for that in this course, even if you cannot derive everything that you've seen on the boards later on. That doesn't matter. You can read papers and books for that, right? But yeah, lose your fear and don't hesitate. And work with somebody who's not on your comfort side. That's very helpful. All right, any questions? Yeah.
There are overlapping challenges. So also there are policies. If there is synonymous mutation at one side, it may be possible that it will affect other.
Some of them, but most of the experimental data we have, that's not the case. I mean, not just my lab's data, but other people's data as well. It's not usually in these kinds of situations where there's overlapping genes and so on. So yeah, that doesn't explain it always. You had a question? What's also changing? DFEs are changing. Yes, yes, yes.
So far, I've seen the focus always like, okay, what's the DFE, but the DFE is a function of time. But probably it's happened at very slow time scales.
No, so the question is DFEs are also changing over time. So what do you have to say for that, right? Like you're just showing DFEs at one single time snapshot. So that's true. DFEs will change over time. There are a few studies now showing how DFEs change over what time scales, by how much, and so on. Not too many as far as I know, but there are certainly. And even in our case, in some sense, you can think of DFEs not even across time, but across environments, they change, right? So the data I showed you were from the same set of mutants tested in different environments. And in those 16 environments, the DFEs look different, right? I mean, they're not dramatically different, but they are a little bit different. So, and this should not surprise anybody, correct? Because we know that the effect of a mutation is dependent on the environment. So any mutation in that whole DFE is sitting in that position in environment A, but may very well flip to the other side in environment B, correct? So DFEs are certainly not static, they are dynamic, and a large part of what I think maybe next week you'll talk about is how those DFEs are changing over time as adaptation proceeds and so on and so forth. But the mystery still remains that across like all these different studies with different organisms, with different environments being tested and so on, there isn't a very consistent pattern of under what situations are DFEs more right shifted versus more left shifted. Some patterns exist. So if you have DFEs measured in completely bizarre environments, or for this Arabidopsis study that I showed you earlier, the evolution paper, they measured Arabidopsis populations, MA lines generated from location A versus location B, and then transplanted them at different locations. They found that the DFEs were typically more beneficial if they were away from where they had grown up, right? Where they had presumably small microclimatic differences were important there. But that kind of pattern is not consistent across lineages, doesn't hold up always, and so on. So some of the patterns make sense, but some of them don't necessarily make sense. So we're missing something else, some other factors that we don't fully grasp. I should also say some of the differences that we see across environments or over time may also be a result of sampling size. So when we have increased sample sizes for a more recent study that we did where some of the same mutants were included, we find that the differences across environments in broad patterns sort of become much smaller. So yeah, there are many, many problems that we don't fully understand yet. Yeah. Sorry, can you repeat?
Like we do get a synonymous mutation as peaks and association studies, but a lot of the time, like they are in LD with some other mutation or other way. So what is the probability like if the synonymous is actually working or is it?
That's a good question, and this would have this is what people thought about in the early days of GWAS studies, where they started finding synonymous changes that were also associated with the phenotype of interest, which is why people did experiments then in the laboratory to ask, if I only make that synonymous change versus not, what do I find? And in many of those cases, they found that the synonymous change is responsible for whatever change that's observable and measurable. Whether it leads to disease phenotype is a slightly different problem, because you can't address that in a cell line, because the disease is manifested in a human in a complicated way. But at least there's a phenotype. And in some cases, like you suggested, there is interaction between a synonymous and a non-synonymous change, and both of those are important and required to explain a phenotype change. So synonymous changes in some cases, well-known documented cases of where alone the synonymous change has no impact, but in conjunction with another non-synonymous change, it does have an impact. So yes. Okay. Yeah.
So also, like, if it's an asexual equalizer, then it's a different thing, but the organism. Then apart from the environment, there can also be how it reproduces or how other things that can affect what sort of mutations it carries. Like if it's I don't know, the cell thing, or then there can be some or I don't know, outcrossing certain. These things maybe they can affect. Like for specific cases, then you can explain what sort of things you're seeing. Maybe not the general.
I'm missing something in your question. Can you tell me? So you're saying that the kind of mutations you're getting changes the DFE, or you're saying something else?
I'm saying that the DFE that you are observing, that you can explain based on what a specific organism, how it reproduces or something, right?
How? Maybe think a little bit more, and
Yeah.
because I would love to know any other hypotheses. Yes. Yeah. Okay.
And just one point about this, they may like when the during the fitness are measured with immediate background or
Immediate background. Yeah. In most cases, the mutants we have are single mutants compared to the original ancestor. So it's the same. But in some cases, we have like second or third step mutations. And so we compare those to the previous step as the ancestor, just as a way to increase sample size. Because as I said, this experiment takes like 3 months to do. It's tedious as hell. And you have to have so many lineages to get even samples of 100. And Shazia has done some of these experiments. You can ask her how annoying it is to keep up with it. Anyway, yeah. Because the mutation rate is small, or not as high as we would have liked. Yeah. Yes. What about it? You mean affected by synonymous changes? Yeah, so there's good evidence for that as well. For DNA structural stability, there isn't very good evidence. But for RNA, it's clear that it does make a difference, because it can change the base pairing as you fold. So you saw in Supratim's talk, for example, the other day, a small change in which bases are there will change your folding, right, of the mRNA. That is known to have a big impact. And so some of the effects in particularly E. coli at the 5' end of genes that are observed with synonymous changes are attributed to mRNA structure and stability. So structure becomes so stable that it's no longer able to be unwound for translation to happen. And that becomes a problem. Sorry? Yeah. Correct. So I'm saying that does have an impact in some cases, but it doesn't explain a bunch of other cases. So in some of our mutants, for example, we looked at predictions of mRNA folding structure and stability, and many of them were not explained by what that was, or there wasn't a big change, but there was a big change in fitness. But some of them did seem to be affected by structure. mRNA structure. Yeah. Yeah. Yeah.
Are there specific codon amino acid pairs in a particular protein? Does it depend more on, for fitness, does it depend more on a particular codon amino acid pair or the position of the amino acid?
I'm not sure I understand codon amino acid pair.
So, if there is one particular amino acid which is present at different positions, would a particular pair be more important? So, one particular codon at all instances of that amino acid, or will one position have one codon or the other?
Yeah, it's the latter. So people have asked exactly this question where said, for example, for valine, let's just change all valine codons and, ask if the change matters differently at different positions where valine is present. And it does have different impacts. So valine at position A may be encoded by, I'm completely making this up, GGA, but at another position, maybe GGG. And maybe the GGG doesn't matter there, but if GGG is here, it matters. So it is very much position dependent as well. And that also indicates that it's not a codon bias effect broadly, but it is position specific, maybe something to do with mRNA structure, something to do with some motif which is required for regulating translation or transcription. So the problem is that as organisms have evolved, there are so many layers of information that are plastered onto one poor DNA piece, right? That there are many, many things that can break down if you change one base here and there. And so a lot of what I suspect is happening with synonymous codon changes, the differences and variation we see across organisms, has to do with which information content is now being disrupted by that particular change, and which of those is much more relevant and important for fitness and phenotype in that organism. And the answer to that varies across organisms and, of course, across the genome. So, yeah.
So, I don't get why we would why should we think that most mutations should be to begin with? In the sense that, like two ways if you approach, like if you are thinking in a way that over evolution, whatever is useful or have been accumulated in a particular way, then we would expect that most mutations would be different. But if you think about evolution the other way, that a lot of things are happening, and in that sense, most of the things that are harmful would have already been weeded out.
But they can come back, right?
Yeah.
As in, yeah.
Yeah, I mean, then they can come back. So, but in some sense, what would be more plausible or more room will be there for things that can keep happening to and fro, which don't really have much consequence.
So you're saying neutral mutation will be much more, yeah, so we're ignoring neutral mutations in what the thing I discussed, yeah.
Which could have been neutral mutations at certain instances, but may not be neutral mutations anymore.
Right. So, okay, so I think you'll learn more about this much more formally and much more like with the maths and so on and so forth. But you're right on both counts, I think. So the intuition is that as organisms evolve in a certain environment and as they get better and better over time, as they adapt, there are fewer and fewer beneficial mutations available. Many of the ones that were beneficial have been fixed already, whereas the deleterious ones, if they are being purged from the population, they are now available to happen again, right? Because in the sequence, they can happen again. And so there are more deleterious mutations than that are available. So that's one reason why we might expect that. But a reason not to expect it is perhaps to say, okay, well, if environmental change is very frequent, right? You're constantly encountering new environments, then you constantly have a new set of beneficial mutations that might be available. And that's where pleiotropy becomes important, right? If there are beneficial mutations that are broadly beneficial, then in some sense, you've already fixed those mutations while adapting to environment A, and many of those are also kind of beneficial in environment B. And so that pool is not changing too much as you shift across environments. Of course, if you shift across very different environments, then yes, a new pool becomes available to you. And the second, what was the second thing that you said? That
that if it was, if the situation was such that we were not, I mean, we were the evolution happens in such such a way, I mean, it can happen together also, both processes. That over time.
Neutral mutations can now become, yeah, and that is observed as well, right? That previously neutral mutations can have impacts on fitness later on. Does happen. But so the peak is still expected in general to be neutral, right? And that is observed in many of the DFEs. The peak is somewhere near neutral or near neutral. But comparing deleterious versus beneficial mutations, we see many more beneficial than we would expect compared to deleterious. And that was the, yeah, so that's, thanks for bringing that up. Yes. Yeah. Yeah, so you remember that one paper I showed in Salmonella in my first lecture 2 days ago with RPSB gene mutations. So that study was actually meant to ask for DFEs for synonymous and non-synonymous substitutions. And in that gene, it's an essential gene, ribosomal protein, very important. They found that most mutations were again deleterious, that was synonymous as well. In that particular gene as well. So that's actually a bit, yeah, that was one of the first studies properly showing this.
So the discussion just was in context of an environment, like in context of an organism already somewhat adapted to an environment. So if an organism is put in a new environment, what are the fractions of deleterious? Do we expect to observe, given that there is no selection pressure on?
Yeah, so that's kind of what I was discussing earlier a little bit, that in some cases, so the problem is that we don't know exactly what environments organisms have seen in their evolutionary history. We can sort of guess a little bit. When people have tried to measure DFEs in 2 different environments, one of which is presumably something they have not seen before, a big stressful situation, there, if you impose stress of some sort, you often find that the fraction of beneficial mutations increases, as you'd expect from this logic. But it doesn't always pan out in cases which are a little bit more gray, where I don't know how new this environment is for E. coli, for example. So in those cases, if you use the ancestral fitness in that environment as a measure of how adapted you are or how much you've seen that environment, and you ask if there's a correlation, there isn't always a correlation. Sometimes there is, but sometimes there isn't. And that is what I was saying. We don't, so it doesn't always make sense or match our prediction, but sometimes it does.
I was just, and some of the thought process behind trying to ask this question is, in a fitness landscape, are there by default more positions where it can go deleterious than beneficial?
Okay, so one,
Like by default, if we do the math, like on a preset, if there are more positions which are more likely to be deleterious than.
so it depends on where you start in general, but maybe you guys have something to say about that. Has somebody measured this? So question is, in a random fitness landscape, if you start from any random position, are there many more deleterious mutations likely or beneficial mutations likely? Do you have an answer for that? No.
Typically, they are not random. There is some correlation. Random is actually an approximate, very limiting case. But if you have this correlated ones, there you can sort of, I think, make some predictions about how much action.
Yeah, I think the question was more about like there's a random fitness landscape, what would you expect to see? And so there I feel it's very hard to give an answer because it really depends on how many peaks and where your population is starting and so on. So, question? You mean mechanistically what is happening? I don't know. For some of them, I can, because we see a correlation with the structure of the mRNA after the initial mutations and then with the new mutation, can hand wave and hypothesize that maybe that's what's going on. But for some of the other variants that we found, that was not explaining anything. We also had a second hypothesis based on work that other people had done before that in the mRNA, if there is motifs that bind to ribosomes, similar looking to the ribosome binding site at the translation start site, then those RBS-like motifs will also stall ribosomes in between and reduce the rate of protein production. So we had some correlation with this initially as well. But in the evolved lineages, that doesn't pan out either because those sites were not, those motifs were not changed. So, yeah, don't know.
Among the amino acids which are encoded by multiple codons, are synonymous mutations in codons associated with certain amino acids more likely to be less neutral?
In some amino acids are neutral, synonymous changes more neutral than others. I don't think we have a very good answer for this generally experimentally. Comparative data, yeah, there are some, you can suggest that those are probably more neutral. So you find, for example, the number of synonymous substitutions you find relative to what's possible does vary a little bit across amino acids. But yeah, I do. Yes. What leads to a mutation are happening in the first place? So typically two sets of processes. One is that there's damage to the DNA. This could be due to UV, oxidative damage or something inside the cell. So if there's oxidative radicals that are generated, which happens normally, with enzymatic reactions in the cell, these can go and damage the DNA, and that causes a mutation. You can have double-strand breaks that happen also because of mutagenesis, where your double-stranded DNA basically has a break that comes up. And in the process of repairing it, there can be mutation. And so, yeah. And then a second set of mutations happen by the machinery that fixes the problems that are. So let's say there's damage due to UV, and there is oxidation or deamination of a particular base. Then the repair machinery that comes in to fix that problem, sometimes that is error prone, and that introduces new errors. So that there also mutations happen. And then, of course, the corollary is that if you don't have some of these DNA repair machinery functional, maybe because you got a mutation that caused loss of function of that machinery, then that mutation doesn't get repaired, right? Which happened. And maybe that machinery would have fixed it, but now the machinery is not working. And so, yeah. All right. Oh, okay.
Suppose if there is a mutation gene is expressed normally, so will that also undergo selection?
What will undergo selection? There's a regulatory mutation,
Yeah,
but there's no expression change. You tell me, actually. I think you've learned enough in this course already to answer that. Will it be visible to selection? No. If expression is the only phenotypic change that is possible and relevant, could it have other phenotypic effects by other mechanisms, maybe the folding of that area? Sure.
Suppose it is changing the label of expression on stoichiometry.
Of? Oh, stoichiometry of that gene product versus something else. Yeah.
Then it should undergo selection.
It could, right? Because that phenotypic change may not change fitness. If it doesn't change fitness, then it's not visible to selection. Right? Make sense? You're introducing new and new cheating variants to this problem. Yeah. Yeah. So if there are multiple copies of a gene, if there is a functional copy that does the job, then maybe this one doesn't have an impact. But there are cases where it can have an impact because it goes and binds to something it's not supposed to bind to, right? So I can give you these kinds of hand-wavy answers because there are, empirical data that support any of these. But it's hard to predict a priori which of these possibilities is necessarily going to happen without having some more data. Yeah? Okay. All right. Oh, okay. Yeah.
This experimental experiment, we are in users because of transcription plan, like replication errors.
So in our studies, we have not added any mutagenesis agent, but others have added it. So EMS is mutagenesis agent that's very often used in experiments. So the type of mutations that occur naturally versus with mutagenesis are different very often. And so if that is of concern or of interest, then that changes something. What is the effect of? But the mutations are changing genotype, no? I don't think I'm understanding your question. Are you able to understand? Oh, you're saying, is that because of plasticity? Yeah, we would say it's a form of plasticity, right? Because the genotype is the same and you're asking for fitness effects in different environments and they may be different in, yeah. Thank you.